going to talk about two-dimensional electronic spectroscopy for ultrafast spectral diffusion and excitation energy transfer. So, floor is yours.
All right. Thank you, Thomas.
I hope you didn't change the title, right?
No, I didn't. I am sticking with my old title. So, all right, do you see it?
Yeah.
Okay, but I slightly changed the order of my talk, which is different from the title. Anyway, thank you, Thomas. Thank you, Maxime, for putting all of this together. It's a great opportunity to really see everyone, although it's virtually. Hopefully, this will change next year sometime. So today I'm going to talk about some applications of 2D electronic spectroscopy that we have been doing in Singapore. And essentially, wait, it's a bit stored. Okay. So it's going to be broken into two parts, vaguely related to each other. First, I'll talk about some excitation energy transfer processes in plant photosynthetic systems. And then I'll talk about more at the molecular level, talking about spectral diffusion dynamics of chlorophyll system, and in particular, looking at some dative bonds fluctuation dynamics. So first off, I should thank the people who really did all the work and our collaborators. So I should give me 1 second. Okay, so this is my group and alumni, and the bulk of the group, the stuff that you're gonna hear is spearheaded by my excellent student and currently postdoc Nyert, together with the other members of my team, my research group, and our samples are provided by our collaborators from Hungary and France. In particular, the Hungarian group led by Peter Lambrev. We've been working together for a long time, and it's not just samples that he's providing, but a lot of like in-depth discussion and also experimentation and so on and so forth. And we have like great help from our theoretical friends from the University of Groningen, the host being one of them, and also my colleague from NTU, Yun Peng, provide also theoretical help. So I'll talk about the light harvesting and plant photosystem 2 first. So this is basically the whole big machinery of the plant photosynthetic machinery. And it's basically the whole thing churns out, takes in light and then churns out ATP and the energy molecules. So there's two parts of it, the photosystem 2 and photosystem 1. So we're going to mainly talk about just photosystem 2. And photosynthesis 2, most people will note it as basically responsible for the famous water splitting reaction. So essentially, light gets absorbed by all these light harvesting complexes and directed into the reaction center where the water splitting reaction happened. So the questions to ask is basically, what is the energy pathway and network that connects all the excitation energy transfer to the reaction center? What are the routes that all these energy passes through both intra-subunit and inter-units? And is there certain transfer mechanisms or design motif in all these transfer processes? For instance, for efficiency and as well as photoregulation. So this big photosystem 2, the structure has been solved by cryo-TEM not too long time ago. What I mean is a super complex has been solved a few years back, and this is how it looks like. Briefly, I'll explain how the structure is and what it does. So this whole thing is actually what's called a C2S2M2 super complex. In it, it's actually a dimer, and each monomer, each block of it, contains the reaction center. This whole thing is the core, and attached to it are the light harvesting complexes. Okay, so over here you see there's light harvesting complex 2, LHC2. These LHC2s basically contain more than half of the pigments in the complex. So essentially they bear the brunt of like collecting all the photons. The whole thing basically contains about 300 chlorophylls, 90 carotenoids, so it's a big monster collecting light and directing it into the direction center. Apart from the LHC2, there are also kind of like each LHC2 is actually a trimer, and there are also like minor light harvesting complexes, CP24, CP29, CP26, which basically are similar to a monomer of LHC2, plus or minus a couple of like chlorophylls and carotenoids. So essentially, the whole aim is to try to figure out where the pathways, like kind of like where are the road system that connects all the energy transfer processes. So of course, this is very complex, and you know, up to now, there are some studies using 2DES to look at subunits of these huge complex, including the core complex system, and most of the efforts are directed at LHC2 because it's the most abundant and you know, and more easily available. So just a quick summary of what has been done in terms of 2DES or looking at the energy transfer processes and related stuff in these subunits and units of plant PS2. A lot of work, some beautiful work has been done looking at the energy transfer processes and also the charge separation processes in the photosystem 2 reaction center by the group of Ogilvy, Van Grondel, and Dwayne Miller. But as I say, most of the stuff was done on LHC2, and there was some work recently done by looking at the carotenoids absorbing and then transferring over to the chlorophyll. And there are also quite a bit of work that has been looking at LHC2, the chlorophyll state. So the chlorophyll, the LHC2, basically the spectrum looks like this, right? So each monomer of LHC2 contains 8 chlorophyll A and 6 chlorophyll B. So this part of it, the slightly bluer part, are the chlorophyll B, and this is the chlorophyll A. So these sticks here basically kind of is a representation of the excitons that results from the coupling between these 14 chlorophylls. And the earlier work was mainly looking at how the energy transfer from the higher energy chlorophyll B to the chlorophyll A. So those works includes 2DES studies as well as 2D electronic vibrational studies. So what I'm going to really talk to you about is the energy transfer process or equilibration process that happens in the chlorophyll A at the lower end lower band of the LHC2 system. So these LHC2, the chlorophyll A portion of it, right, has about 8 states. So now, if our fundamental aim is to try to understand the energy flow from LHC2 to the other parts of the PS2, the consensus is that it is using the chlorophyll A manifold, the states from here, that do the transfer. So understanding the excitation energy transfer network of these parts of the LHC2 will be quite important. Now, the problem is there's a lot of states over here, and these states are very closely packed together within these about 20 nanometer bandwidth, right? So it is to be expected that these states will have like equilibration dynamics, meaning that there would be downhill and uphill energy transfer. This is to be expected because this bandwidth here at room temperature is roughly about 1.5 kT. So you could expect that some of the energies will actually flow uphill. So we're thinking that if we really want to get the EET network, the energy transfer network for LHC2 as well as the whole big PS2 system, we better be able to figure out all these minute uphill and downhill processes. So essentially, that's what we had done a couple of years ago, looking at these uphill and downhill energy transfer processes. So if you look at the blue slice and the red slice, and if you take them out, they are kind of like quasi-narrow pump broadband probe transient spectra. Then what you can see is that from the blue pump, the slice that you pump at 665, you actually see this downhill trend. So this is to be expected and really nothing new. But you can also look at the slice that is in the red, and you can see that there's the trend towards going to higher frequency, shorter wavelength. And these you can clearly see that it's attributed to uphill energy transfer processes. So with this confidence in being able to see uphill processes in very tightly, very closely, densely packed excitonics levels, we then try to uncover the transfer network of the chlorophyll A part of the whole LHC2 system. So these are so the idea is that if you have a 2D spectrum, a couple of years ago, we worked out by Tobias Pixner group, and also applied to FMO system by Donatus Zigmatas group. You can see that from a 2D spectrum, you should be able to extract the information that connects all the different rates and the rate transfer matrix that connects the different excitonic state. So we make use of this idea and try to get at the transfer network of LHC2. Unfortunately, the LHC2 system is slightly more, there's more states closely packed than compared to FMO system. So it's kind of difficult to actually just have a clean inversion. So what we need to do is actually to modify the algorithm a little bit, which is to actually introduce a kind of like a fitting routine in order to extract the energy level and as well as the transfer matrices. And then from there, try to compare with theoretical models and then make assignments to these states and how they transfer the energy to each other. So that's what we did. So we acquire high quality signal at very low energy, pulse energy, such that there is no singlet annihilation condition, no singlet annihilation processes detected. And we run a whole series of TW up to about 100 picosecond. And then what we then do is try to guess what is the 2D, based on, to build up a 2D spectrum based on a transition dipole matrix, a transfer rate matrix, and a species associated spectra. So it essentially tries to generate a 2D spectrum by basically putting Gaussians onto, basically build a 2D spectrum with two-dimensional Gaussians to make up the diagonal as well as the cross peak. Okay, so then you can then use this to try to fit to the experimental data using an iterative fit. So if you do this well enough, it will converge, and this is basically the fit results that we have. And from there, the results from the transfer matrix and the states that you recover, you can then build up the network system where this energy transfer process happens. So this is basically the results of it. So what we have is the phenomenological fit obtaining all these energy levels and what are the different rates that can transfer to each other. So at this at this point, you can't really kind of assign it because these are just spectral components connecting to each other via different rates. So what you can then do is look at basically theoretical models, for example, Van Grondel or Rangel or Zucchelli models, and try to see which of these states correspond to the calculation, the assignment. So if you do that, you can kind of get an idea of what the assignment is. And then you can then take a look and then see what are the main features that you actually can get out of all this network system. So one thing we sort of notice is that there is 3 states at the lowest energy levels, and they are basically disconnected from each other. So that's kind of surprising because one would think that if energy were to flow, it would basically all flow down to the terminal state, and then from there flow on to the CP29 or CP24 LHC system before going into the reaction center. But in fact, what happened was there are 3 energy levels that are very low, and they are basically rather unconnected from each other. And essentially what it then, if your energy gets stuck in some of this level, what happens is that it has to rely on uphill energy transfer process to actually surmount that barrier to then get to your terminal state. or for that matter, to other states. So this kind of phenomenon of having low energy states which are not connected to each other has kind of been reported in other areas as well. One such report was also quite recently that using Howe burning experiment, they see actually this kind of like low energy levels such as this. And also there's also calculations on PS2 core, not on LHC2, but on PS2 core, that basically observe that there are actually low-lying energy levels that are actually lower than the reaction center states, that basically can only provide transfer to the reaction center state, the charge transfer state, by uphill energy transfer process. So it's a question that can be asked whether this kind of disconnected low energy level pool is a kind of like a design motif in all these network processes in this LPS2, or for that and for that matter, or for the photosynthetic systems. And what are the really functions of all these states, these disconnected states? So we have also tried to actually look at the network systems of other LHC states. For example, we also tried to look at the transfer network of this kind of marine green Bryopsis corticulans, a kind of marine green algae, which has slightly different proportion of chlorophyll A and chlorophyll B. And it turns out that it's also observed that we see low energy, rather weakly coupled, weakly connected low energy states. So it will be interesting to look at other systems to see whether such kind of like phenomenon also exists for other subunits of the photosystem too. Which brings us back to looking at more recent work, talking about more recent work that we've done. This is COVID era work done sometime this year. So we move on and try to look at bigger subunits of photosystem 2. So from our collaborators from France, Stefano Caffari, he's able to actually tear out different subunits of these PS2, and then we can then run 2D spectrum on them. So one of these things that we are now performing 2D spectrum studies, spectroscopy studies on, is this system where it is a LHC2 coupled with a CP29 and a CP24 subunit. So the idea is basically eventually to look at different subunits, different combination of subunits, and try to figure out what are the energy transfer process between them. So why do we choose this? Well, one thing is because it's available. Another thing that's of interest is actually that it's believed that the major energy transfer pathway from the light harvesting complex into the reaction center is actually via connections with CP29. Before then, it then goes to the reaction center. So it'll be interesting to actually look at the 2D spectrum of these LHC2, CP29, CP24 system, and try to locate the energy transfer nodes where energy, to see whether we can see telltale sign of that energy transfer process. So to zoom it up into this system, the recent structural determination actually has certain kind of like suggestion where all this transfer could be. So this is from the paper of Suma and Wei of the structure of PS2 by cryo-TEM, and you can see that some of these chlorophyll A's are actually rather could be the candidates for the nodes where the transfer process actually happened. So we then did the 2D spectrum at room temperature. So these are actually what's called a 2D decay associated spectra. All right. So we break down to basically the transfer processes of different spectral components at the different time scale. The first two are the sub 100 femtosecond and the sub 500 femtosecond dynamics are rather similar. There are some differences that we are still analyzing. But what is actually interesting is actually looking at the 3 picosecond DAS 2D spectrum. And from there, you can actually see that there's some differences in the 2D DAS spectra at this cross peak here at about 648 to which is basically the cross peak that connects 648 nanometer to 668 nanometer. And this may suggest that we're actually seeing this cross peak dynamic difference between the dynamic difference between these cross peaks of I didn't really mention. So this top 3 spectrum is actually for the trimer, LHC trimer, whereas these bottom 3 are for the LHC to CP2429 system. So for this bigger system, there is this extra cross peak here, and this may be indicating that that this is actually reflecting some of the transfer processes that's happening between LHC2 to CP29. So this is preliminary data, and we're still trying to analyze it more. So basically, a quick summary for what has been talked about. We looked at LHC2, and there are disconnected low energy pools in LHC2, and recent preliminary data on PS2 looking at LHC2 CP2924. There seems to be some suggestion that we're actually looking at some inter-subunit energy transfer nodes. So now we go on to talk about the other topic of the talk, which is basically looking at the spectral diffusion studies of chlorophyll. And the work is actually mainly done by my postdoc, Nyert, and Long and Jamie, who are my graduate students. So, so switch gear now to molecular system. So I guess like a quick kind of slide on to basically talk about what spectral diffusion. So on the 2D spectrum, the peak shape changes with TW basically tells you a lot of information about spectral diffusion process. So essentially, at short TW, you notice that your 2D peaks will typically be more elongated, and it goes on to be more spherical when it gets rounded. So this process of like slowly becoming uncorrelated tells you the process of spectral diffusion. Now, how can you analyze it? One way to analyze it is actually using this method called the central line slope, where you try to pick out the maximal lines as you draw slices and then connect them out. It turns out that if you churn out the equation, this gradient here is proportional to what's called the frequency fluctuation correlation function, which is the quantitative measure of spectral diffusion. So this was kind of like developed by the Fair group for 2D IR, and now more recently, more and more usage in the electronic spectroscopy as well. So our interests are in looking at spectral diffusion is related, of course, to the things that we are working on in the light harvesting complexes, since chlorophyll molecules are the major components of them. So we started to try to look at their spectral diffusion to try to understand the, how the environment actually affect the dynamics of the the spectral diffusion dynamics of chlorophyll. So this is a typical spectrum of a QY band absorption of chlorophyll in different solvents. This gray part is actually our our laser pulses. So you can see that this is a 2D spectrum of the QY band of chlorophyll A in pyridine as the CLS changes with time. You can typically then fit them to a multi-exponential function, which basically tells you the normalized FFCF. All right, and these are some examples. These are basically the chlorophyll A in different solvent, in THF, in diethyl ether, and in methanol. You can fit it typically to 3 decay components plus a constant offset. And then from the different components of the decay, you can then try to assign what are the processes that actually are responsible for that. So typically, the offset is due to static inhomogeneity, all right? Structural inhomogeneity, long-term, essentially very extremely slow spectral diffusion. The fast component, the sub 500 femtosecond component, is usually attributed to some kind of a motion, small amplitude motion of the solvent, and the 2 to 5 picosecond is usually attributed to viscosity of the solvent. For all these solvents that we try, these numbers are not too different. What is actually quite different is actually the third component, which is the tens of picosecond component. So for example, in this case, for methanol, it has a very slow 40 picosecond component, which is lacking in the other two solvents. So, and these usually you can attribute to solvent rearrangement. And in this case, because it's methanol, it has been kind of like assigned to essentially the hydrogen bond network hindering the rearrangement of the solvent molecules, hence giving rise to this 40 picosecond. So we started to then look at more comprehensively chlorophyll A in all sorts of different solvents and try to see whether we can identify other molecular processes that basically gives you all these spectral diffusion components. So we started to play around with aprotic solvents because we want to not have to consider about hydrogen bond. So we put chlorophyll A in all sorts of different aprotic solvents with a whole range of different pKa values. That means different basic CD. And interestingly, when we come out with the results, there seems to be some correlation with the basic CD with one of the with the third component, the tens of picosecond component. So if I just expand it out, you can see that as the pKa increases, namely meaning the basic CD of this solvent increase, there is also a corresponding trend of increasing of the tau3, the decay constant, as well as the amplitude. So then we think about it, what can give rise to this? Obviously, the strong case is that these solvents are all forming coordinated complex with chlorophyll, with the magnesium center of your chlorophyll. So this is basically a THF linking up with a chlorophyll. And essentially, if you have a stronger Lewis base, you will expect that these coordination bond is essentially the dative bond given by the solvent to the magnesium center. Given stronger Lewis acid, there should be a stronger dative bond. Hence, we form the hypothesis that these numbers here, the amplitude and the this component, this A3 and tau3 component, the decay component, is actually measuring the dynamics of the solvent ligand dative bond dynamics. So to try to see whether this hypothesis holds, we did the same experiment on an analog molecule, the chlorine E6. Essentially, it's like a chlorophyll, except that there is no magnesium in the middle. So the idea is that if there is no magnesium in the middle, what we think as the dative bond dynamic should not appear in this system. And indeed, that was kind of what happened. So if we do the 2DES on chlorine E6 in the different solvents, THF, 3F pyridine, and pyridine, although there's a huge difference in the basic CD, the tau3, the time scale of these the decay process, basically did not change at all. That is in comparison with chlorophyll A and chlorophyll B, which I've already shown earlier, that there is a huge difference, right? So that seems to really indicate that this tau3 really depends on magnesium ion, and hence can be attributed to the solvent's lone pair interaction with the magnesium ion in the chlorophyll molecules. So what about the amplitude, the A amplitude constant? This A constant is actually related to the fluctuation amplitude, right? So essentially this fluctuation and amplitude is essentially this bandwidth that the frequency can fluctuate as my molecule jumps about. Sorry. All right. Okay. As it changes, it's like a fluctuate as the bond fluctuate. You would expect that the HOMO and LUMO orbitals around of the porphyrin ring to change and be perturbed, and these then give rise to your chlorophyll QY transition frequency fluctuation. So in other words, if the hypothesis is that if this this fluctuation actually tracks the strength of the magnesium ligand dative bond. All right. So in order to try to show whether this is true, we did a series of calculation, a kind of like proxy calculation, basically to look at the difference in the transition frequency of Qy of the difference between a hexa coordinate and a penta coordinate chlorophyll. Okay, so basically, if we do calculate these using TD-DFT, the energy, the Qy transition energy between the penta and the hexa, these delta E should kind of track the delta tree that we have like measured, and to see whether there's actually some correlation. It turns out that there is some correlation. So basically, these are the different data points of the chlorophyll A of the different solvent. The experimental recovered delta tree plotted with delta E, which is this calculation, and you can see that there is definitely some correlation. So that seems to basically support the hypothesis that we posed earlier, that the delta tree is actually a relative measure of the strength of the magnesium ligand dative bond, and tau tree in this case, because it actually the delta tree basically is fluctuating between the strong, essentially akin to breaking and forming of the dative bond. The tau tree is actually measuring a bit like the bond breaking and forming of this dative bond. So what is the significance of this study? So basically, this is a direct way of looking at the dynamics of a molecular level process, namely this of the dative bond. And this may be important in you know, like in kind of like chemical application. So of course, like the dative bond is like very much related to Lewis acidity and basicity, which is of course one of the most important chemical properties. So if like we can basically design some experiment to actually look at all these dative bonds to try to understand more about Lewis and basicity of different you know, like interaction. And also a lot of metal center catalysis in chemical synthesis, such as Wilkinson catalyst, the active site actually relies on all these ligands coming into your metal center. So if we can devise way of actually looking at the lability, the dynamics of this lability of all these ligands, it may be of use to chemical synthesis. And you can also use this CLS analysis probably apart from just looking at dative bond. There will be other molecular processes that you can actually look at if you can carefully design some control experiments and look at the correct system. So with that, I kind of like end my talk, and this is basically the summary. And I think I'll stop here, and I'll be happy to answer any questions that you guys may have. Thank you.
Thank you very much. If there's any questions, please just drop a note in the chat or raise your hand. Okay, I think I could. So I have a question regarding these low-lying pools, because you didn't really give explanation of why they're there. Do you have an idea why you would have these three different pools and what their function would be?
We don't have any, but it has been kind of like observed and then speculated for the reaction center that there are all these low-lying pools that relies on uphill energy transfer process, and in this case, in which case these would be slower energy transfer processes. So they may be there for kind of like a speed bump when there's too much energy floating around. There's speculation that that's the reason why, if any, that they may be there. But currently, what we know is what we can know from our experiment. We do not know the reason. We only see them there. So I have really no clue why, conclusively, why they should be there. Maybe you can help me.
Well, hopefully we'll learn something. So Daniel Turner have a question. You're free to unmute yourself if you want to ask it in real life.
Hi, thank you. Very nice talk. My question on your second part is, how did you account for the dynamic Stokes shift of the stimulated emission peak
Oh,
when you're analyzing the peak dynamics?
okay. Essentially, the Stokes shift is very small. So and if you do the CLS analysis, essentially the Stokes shift doesn't really contribute, doesn't really, in a sense, how should I say, interfere with the CLS analysis that much. So we don't really account for it. Essentially, the effect is very small, so it really doesn't matter.
It just
the way
in
that
this,
we analyze
because
it.
in this sample the Stokes shift is so small, you
Yes,
just
so
ignoring it.
you can actually see it. Let me see if I can go back there to show you.
I thought you showed the fluorescent spectrum and it was a large Stokes shift.
Oh
But
no,
maybe
I didn't show
my
any
memory was
fluorescence.
off.
So sorry, this is a bit slow going back. So you can see here, there is some Stokes shift. You can see this bump here moving to the left a little bit. Okay, but if you do the CLS by slicing this way, the Stokes shift effect is not that strong.
Great.
And
Thank
in
you.
any case, these are not these are basically absorption spectrum. I didn't put any fluorescence here.
Very good. Thank you. It was a great talk.
You're welcome. Thank you.
So maybe I can follow up a little bit on this question because I'm interested in that. So essentially, the shift from the Stokes shift, it will not really affect the slope line if the Stokes shift is like uniform for all frequencies, right? So,
that's right. Yeah.
Yeah. Do you have a number, approximate number of the Stokes shift in the centimeters?
I
Not on the top of
kind
your
of
head.
like I can't recall. I could check but I couldn't recall.
I can check it as well probably. But thank you. Other questions?
I have a question, if I may,
Sure.
towards, well, Siang and maybe Thomas to you as well, as you're working together. So all this binding and unbinding, okay, so the time scale and the amplitude, from my perspective, should be relatively easy to be derived from molecular dynamics simulations. Is it so? Have you guys attempted to do that?
That's a good suggestion. I've tried to ask a few people to do it, but I have not gotten any positive answers to do that. So I haven't really asked Thomas to do it before.
Well, so yeah, so since Howe Siang Tom didn't ask us, we didn't try to, but maybe that's an interesting thing to do at a certain stage.
How difficult would it be, Thomas, from your perspective? Is it something, you know, like
in principle,
specific,
it should not be difficult,
or just normal routine stuff that you do like 100 every day?
Well, the point is here that some of these molecules are not as standard as maybe the other thing. So I think it depends a little bit on the details.
Okay, good. Thank you.
Thank you.
Right. We have one more question while we are discussing. Ismail, you can ask your question because now you can talk.
Sorry, I don't know if you can hear me now.
Yes, I can.
We hear you.
Hello. Hi.
Thank you. Very nice talk. I was just curious when you were talking about the tau free time related to this native bond, right? So
yeah.
When you haven't got the magnesium in that structure you showed us, wouldn't you expect to see that component not there at all, or
yes.
a lot?
Yeah, that's a good question. We've been pondering about this, but I guess what we can say at this current, okay, maybe I can like kind of show, basically bring you to that. Essentially asking, okay, if there's no magnesium and if I claim that it's tau3 is actually attributed to the native bond, why am I seeing this thing here, right?
Yes, that's it.
Yeah. So I guess that the whole model we're looking at is kind of a simplified model, and definitely there is strong evidence that tau 3 has to do with the dative bond, but we are not ruling out the possibility that on top of all these dative bonds, there must be some other processes that is of this time scale that happens to all of them. Could be remnants of some, like a longer due to the viscosity or something of the solvent molecule. We haven't quite resolved that yet. But yes, there is probably some other processes. But having said that, if you compare to the, say, for example, pyridine, the contributions, if it exists, is quite small compared to these bigger components.
Thank you very much. Yeah, thanks.
Yeah, thank you. Thanks for the question.
Oh, upon see these nice graphs, another day I was discussing with Dow Wiersma. Well, many of the people know him. The echo peak shift technique.
Yeah.
And it kind of disappeared, okay, from the community now, because everybody began to look at the slope or ellipticity or whatever. But here, well, especially judging from the graph where you just put your cursor upon, okay, this should be quite simple measurement, the echo-peak shift. Did you try to do that?
Well, I was thinking of this. Actually, from our data, we should be able to basically recover the three PEP signal. But no, I have not done it. But they should be similar. Of course, that's by faith. They should be similar.
Well, yeah, that's the whole idea. I was just wondering how similar this would be.
Yep. Okay. I should get some talented undergraduate to do that. Should be doable.
Well, that sounds so easy for you.
Yeah, I'm also thinking that essentially for 3 PEPs versus a 2D, be it CLS or the elliptic city, it would be actually cleaner to do the the 2D experiment simply because you have more signal. You are actually averaging over more points. So the 3 PEPs is just 1 point measurement, right? You look at 1 data point. Whereas over here, I'm taking a gradient over a whole, range of frequency. And in that case, there seems to be more data points for me to average. So yeah, that's what I think. But for academic question is, yeah, it would be good to actually look at the 3 PEPs as well from the same set of data and see whether they're the same.
Okay.
Okay, more questions, guys? Yeah, Thomas?
No, I don't have any further questions. I guess we can discuss again later regarding simulations.
Sounds good.
Okay, so maybe we should thank both Peter and Howe Siang for their presentations, and we hope to see you all in the new year. And maybe we should also say, maybe the Christmas man should say Merry Christmas to everybody.
Well, to those who are involved and concerned, and Happy New Year.
Yeah, thanks everybody, and yeah, we will be looking forward to seeing you again in, what was it, 15th of January.
Yeah, thank you to you for organizing.
Thank you very much.
Thank
you
you
very much.
Yeah.